Right. Hello. Welcome, everyone, to MTRI's third August talk of the summer seminar series. My name is Luca, and I'm a research technician here at the Mersey Research Institute, and I'll be your host and moderator for tonight. Now, before we begin, I would like to acknowledge that the MTRI is located in, and we are meeting from Keswick, one of the 7 districts of Mi'kmaq, home of the Mi'kmaq people. We acknowledge the treaties of peace and friendship, and we thank the Mi'kmaq for their generosity in sharing their homeland with us. Now, for anyone that may be listening for the first time tonight or that might not know about the MTRI, I would just like to give a little background about us very quick. The MTRI is a research-based nonprofit organization located in Southwestern Nova Scotia, close to Acadia National Park and National Historic Site, and within the Southwest Nova Biosphere Reserve. Our mission is to promote, sustain, and conserve biodiversity in Keswick and beyond. And now I would like to welcome tonight's speaker, Rebecca Mader. Rebecca is a master's student of biology in Dr. Alison Walker's lab at Acadia University, and she's also the treasurer of the Mycological Society of America Student and Postdoc Society. Rebecca's seminar tonight will be covering fungal communities in Eastern Hemlock forests and the potential threats these communities face due to the spread of the Hemlock Woolly Adelgid. It's great to have you, Rebecca. And as you know, I've been working a fair bit in hemlock forests myself over the last couple of years, and I'm really looking forward to learn more about understudied community in those habitats. Now, before we begin with the talk, I would just like to remind everyone to please keep your mics muted for the duration of the talk. We will have a dedicated question period at the end where you can ask your questions live. If you do have a question during the talk, you can just type it into the chat window, and we'll get back to that during that dedicated question period. And now with this, I would like to hand over the seminar to you, Rebecca. Thank you very much.
Thanks, Luca. That was a great introduction. Don't know how I'm going to top that for my introduction, but anyway, thank you everyone for coming here to listen to my mushroom talk. I'm, like Luca said, Rebecca Mader. I'm a master's student at Acadia University. I'm working with Alison Walker, a professor at Acadia University, as well as Keith Ager, a retired prof from the University of Northern British Columbia, Matt Smith from Parks Canada, and this presentation will also include a bit of the results of Olivia Crooks, who is a former honor student in our laboratory, who was working on something very similar, so definitely relevant. And with that, let's just dive right into it then. So, as you could surmise from my title, my project centers around the fungal communities of Eastern Hemlock, or Tsuga canadensis. So Eastern Hemlock is a foundation species of Acadian or Wabanaki forest, and it creates very cool, very shady ecosystems, high levels of organic material found in the soils, very acidic soils with low rates of nutrient cycling. So altogether, this creates a very unique ecosystem, and many animals, insects are dependent upon it, and fungi, as we'll soon see. Unfortunately, this ecosystem is currently threatened in Eastern North America by the Hemlock Woolly Adelgid, which I will from now on be referring to as HWA. Hemlock Woolly Adelgid is an aphid-like insect that settles at the base of a hemlock needle on the branch. It covers itself in this waxy coating, which makes it white, like you see in the picture here. And once they've settled down, they start sucking the sap out of the trees. And this can be detrimental to a hemlock tree and can kill it within 4 to 10 years of infestation, depending on the density of this invasive insect upon it. And to add to this, the fungal diversity of Eastern hemlocks, threatened or not, is really not well researched. So there's definitely a research gap in here that my study is trying to address, especially in light of the threatened nature of these trees. So for my project, I had 3 main questions. The first one is, which fungal species actually form associations with Eastern hemlock? Secondly, I wanted to look at if the overall fungal diversity will actually change as the Eastern hemlocks decline. And thirdly, will environmental variables play a significant role in influencing the fungal communities, in addition to Hemlock Woolly Adelgid? So my study area was Kejimkujik National Park. It's a wonderful place, very beautiful, and it's also a great place to study the fungi of Eastern hemlocks because this park hosts most of the old growth undisturbed Eastern Hemlock in our province of Nova Scotia. In addition to this, we also have historical collections that were taken back in the 1990s from certain plots in this park that I'll be using in my present study. So that's definitely an asset as well. Now, one thing I will mention is that although we are technically studying the Hemlock Woolly Adelgid, it's difficult to study the effects of this insect directly because they haven't really been in the park long enough to create severe damage to the trees. So instead, I'm using a proxy disturbance, which is the pale-winged gray moth. This is a native species. However, it can be very destructive to Eastern hemlocks when they have big outbreaks, such as the one that occurred in the early and mid 2000s. This happened from 2004 to about 2006. And the infestation of this pale-winged gray moth severely damaged certain hemlock groves in the park. So this is the disturbance that we will be using to study how the fungal communities respond to disturbances to their host trees. Now let's take a look at the plots that I was using. So this graph shows a picture of Kejimkujik National Park, and it also shows the damage that was done to hemlocks by the pale-winged gray moth. So the light yellow areas indicate that there was a light disturbance. The orange areas indicate that there was moderate disturbance. And then finally, the red areas indicate that there was a severe disturbance to the hemlock trees caused by pale-winged gray moth. And the plots that I'll be using are highlighted here. EMAN plots, both A and B, were undamaged by the pale-winged gray moth. Plot A was lightly to moderately damaged, and then CIMAB2, was severely damaged by pale-winged gray moth. And then plot D isn't quite relevant to this map because it doesn't have any hemlock in it, as we'll explore in the next slide. So this is the exact same map. However, instead of damage, this one indicates the type of tree composition within each of the plots. So the EMAN plots, these both contain old growth hemlock and predominantly old growth hemlock. Plot A contains also old growth hemlock, but also some other conifers such as pine or spruce or fir. Plot D is a hardwood plot, so it doesn't contain significant amounts of hemlock. It's mostly beech. And then CIMAB2 originally was predominantly hemlock-dominated stand back in the 1990s when our historical collections were taken. However, after the severe damage it sustained from the pale-winged gray moth outbreaks in 2002 to 2006, it became more of a mixed plot as other species came in to replace the hemlocks that were killed. So as it stands right now, it is a mixed wood plot. As for my study design, this is just a brief overview of it. So basically, in a nutshell, I'll be taking specimens, whether these be modern field collections or herbarium specimens, and I'll be identifying them using ITS rDNA sequencing as well as phylogenetics. After I get all my IDs down, I'll be able to create a Shannon diversity index value for each plot and each year. This is basically just a number. For those who don't know, it's just a number that gives me an idea of the actual fungal diversity within those plots. And then finally, I'll perform some statistical analysis, make sure I got all my numbers right, and then on we go. So that's the basic layout of my project and the significance of it. Now let's talk a bit about what I've found so far. So my first research question was, which fungal species form associations with Eastern hemlock? To do this, I take a look at the communities of the hemlock plots and compared them with the communities of the non-hemlock plot. In addition to this, I did an extensive literature review, and in that, I found about 175 species which potentially may associate with Eastern hemlock in the literature. Most of these were ectomycorrhizal, and it was great because we saw a lot of those within the plots. If any of you are unfamiliar with what ectomycorrhizal means, it's basically just the saying that the mushrooms or the fungi are able to actually form physical associations within or around the cortical root cells of a tree, and that creates an interface through which the fungus and the tree can exchange photosynthetic sugars and nutrients and carbon and water. Generally, it's considered a mutualistic association. Anyway, so yeah, most of the specimens that we found in the literature and in the forest were ectomycorrhizal. However, many of them seem to be non-specific to hemlock. These are some examples that we found in our plots: Amanita flaviconia, Lacaria laccata, Clavulina coralloides, Lactarius tabitus, my favorite, Leodia librica. All of these were found in Eastern hemlock plots. However, they, according to literature, they don't necessarily associate specifically with hemlock. They may associate with other tree species as well. However, there were some that did associate exclusively with hemlock. These would actually be the ones that would be more threatened by a decline of Eastern hemlock. The logic being that if they run out of host trees to get that photosynthetic carbon from, then they're less likely to be able to survive without them. So we may see a decline in species such as truffles, which have species that are associated obligately with Eastern hemlock. Truffles are an important nutrient source for many animals, like insects, columbola, small mammals. And another type of mushroom would be hydnellum. These have been found to associate heavily with Eastern hemlock, and they're a little less common than your typical mushroom, so definitely a significant part of the biodiversity that we don't want to see lost. And one more thing I wanted to point out, which is not necessarily associated with hemlock right now, but I just found really cool, is that we found one specimen of Sarsaparilla carinthiaca, which, if is true, would be the first record of this species in Nova Scotia, which is very exciting. And we are fairly confident in this because it matches heavily with the original specimen that was submitted to GenBank by the author who described this species. So just something really interesting that I wanted to point out, and just shows that you never really know what you're going to find when you go out to do these projects. Okay, so done question 1. Let's move on to question 2. Will the overall fungal diversity change as Eastern hemlocks decline? To do this, I did 2 different assays, a spatial assay and a temporal assay. The spatial assay was done with the EMAN plots A and B because we don't have any historical data for these. However, they did not sustain any PWG damage, pale wing gray moth damage rather, and they're also old growth Eastern hemlock systems. So with that, we can somewhat assume that these are ecosystems with an intact fungal diversity, which we can then compare to the other plots who had damage and disturbance done in them by pale wing gray moth. So that's the spatial assay. The temporal assay are for plots that we do have historical data from. So we have plot D, which does not contain hemlock. That would be used as a control plot. Plot A had light to moderate pale wing gray moth damage, and then CIMAB2 had moderate to severe pale wing gray moth damage. And by comparing the diversity of the plots back in the 1990s and now, we can get a sense of how the fungal diversity may change in light of disturbance and how it may potentially recover or not after the fact. These are the preliminary results from the spatial assay. I'll draw your attention to the first graph right here. These are the Shannon Diversity Index values for 2021 to 2022 for the EMAN plots and for plot A and CIMAB2. And what we see here is a very high diversity within the EMEN plots, a little less diversity in plot A and CIMAB2, which are the two plots that were damaged by paling gray moth. So initially, we may interpret that as being a decrease in diversity. However, we need to take that with a grain of salt, because in the second graph, which is the number of specimens collected, we also see that plot A and CIMAB2 had fewer collections taken from them. Now, whether that's a bad year or just some other factor, or if it is just a reduction in diversity and abundance, that's hard to tell for now. However, studies go on, and we will dive into that further with the next field season. And now for the second plot, which is the temporal assay. The first plot is, again, the Shannon diversity index throughout the years from plots A, D, and CIMAB2. And again, here we may see a decrease in the diversity because we do see a lower Shannon diversity index, particularly in the year 2022. However, once again, we did also have lower numbers of collections. So similar findings between the first and the second. Mushrooms unfortunately are very temperamental, so it's sometimes difficult to get reliable data without many years of collections and without genetic sequencing of the soil. However, my initial impression would be that there may be lower diversity seen in those damaged plots. But again, take it with a grain of salt. And my third question, which is, will environmental variables impact fungal diversity? To do this, basically, I just took all the plots, and I'll be taking the temperature, precipitation, and soil pH from each of these. We do have some historical data for temperature, precipitation, and pH, not from the EMEN plots, but from everything else. So we'll be able to compare these as well. Now, literature does suggest that all the environmental variables that I mentioned before do affect abundance and diversity. Precipitation, I would say, seems to be the most important factor. However, it's not the only one. For example, we saw the highest diversity in the years 1995 and 2021. 2022 had the lowest diversity, or SDI rather, that we saw. However, 2022 had higher rainfall and precipitation levels than 1995, so there are definitely other factors in play. Change in pH. pH has been correlated with different fungal communities. So, as I mentioned during the introduction, Eastern Hemlock tends to have very acidic soil compared to others. However, when you start getting other tree species coming in who prefer a more neutral soil, that's most likely causing a bit of a change in the pH. And with that, you may have other species coming in. So it's difficult to say. But I do expect to see some differences caused by the environmental effects upon the fungal communities. All right, so that's what I have so far. There's definitely more to do. We have one final field season coming in right now. We've done our June and July collections. We'll be going into, we'll be finishing off August and then continuing into the fall. For these, I'll also be collecting more data, soil pH data, and identifying the remaining specimens. And finally, when all of that's done, we can perform the statistical analysis and get harder numbers on everything that I've spoken about right now. And finally, why should we care about this? Well, biodiversity is key to maintaining productive ecosystems. Excuse me. And fungi are part of that biodiversity, and a very important one as well. The fungal ecosystem of Eastern Hemlocks is really not well studied. And fungi do play a significant role in the health of ecosystems. As well, given the fact that that some of the species are obligately associated with Eastern hemlocks, we may see a loss in certain fungal species. And there may even be more that we just don't know about, again, because this ecosystem isn't well studied. And finally, fungi are just really cool. And it would be a shame to lose anything before we get a good chance to study it. So we should definitely care. And yeah, so with that, I'd just like to thank all of my wonderful funders, everyone I've worked with that has made this project possible so far, and my supervisors as well for getting me through those tough parts and helping me to refine this project a lot throughout the months that we've been working on it. And finally, I know I just said finally, but I'm saying finally again, if you want more, if you'd like to read up on this, we do have a publication coming out soon in the Proceedings of the Nova Scotia Institute of Science. And in that paper, we go through a bit more of the literature and the predictions on how these communities might change as we go forward. And also, if you like fungi and you like to get involved in stuff like this, we do have the 2023 Fall Continental Myco Blitz coming up. We just finished the summer one this week, and we'll be having another one in the fall. So you didn't miss out. There's still a chance. And if you would like any more information on what this is or how to participate, you can take a look at this link right here on mycota.com. Okay, now I'm done. Thank you so much for your attention, everyone, and I'll take any questions that you have.
That was great. Thank you so much, Rebecca. That was a great talk. We will now move into our question period. So for anyone that has any questions, feel free to ask them live now. We'll also be reading out any questions that may pop up in our chat window here in a second. I usually like to break the ice with the first question, so I will start with the first question for you, if you don't mind. How do you take a sample of the actual mycorrhiza? How do you do that? Do you take like a soil sample, or do you,
Yeah, that's a great question. So for this project, we're actually just collecting the sporocarps. That's how the specimens were taken back in the 1990s. So we want to preserve those methods to make sure that it's comparable. So more or less what we do is we just go, we go into the woods and we find a mushroom and we take the whole specimen, take it back to Acadia, and we'll dry it there to preserve it. And then after we do our DNA sequencing and it's identified, it goes into the herbarium.
Awesome. Great. Oh, Keith is asking a question. Keith is asking, Keith Egger, do you have any data yet on pH changes, or is that coming up?
Yeah, also a great question. We have historical data from the master's student who took it during the 1990s. So yeah, we have the pH. However, we don't have pH for many of the recent years because this was something that we added recently. And we just got the meter, which I'm excited to use. So we'll be taking pH for this field season, and we'll compare that to the other field seasons.
Great. Chet, you have a question on your side of on your Facebook side of things?
I do. Someone's asking if you can use environmental DNA sampling in this kind of project.
Yeah. Oh my goodness. I have people in my lab who have been researching environmental DNA, and it is a great technique. However, it's finicky, or at least that has been my impression of it from the people that I've been speaking to. And there's a lot of DNA in the soil as well. We don't know how long fungal DNA stays in the soil, if it's necromass. So it's a great idea, and I definitely would love to try it in maybe PhD or something. But for a master's and for what I'm doing right now, it wasn't entirely applicable, but great question and great technique, definitely.
Perfect. Yeah, we got another big question just follow up. Okay, again, there was a large number of specimens you found in the literature that you suspect to be associated with Eastern Hemlock. If you had to speculate, how many species would you think are specifically associated with Eastern Hemlock stands?
Say that last part again, please.
How many species would you speculate are specifically associated with Eastern Hemlock stands?
Okay, you're putting me on the spot here. So I've read at least one paper that I can't remember the exact number, but it was around 6 or 7 species of Hydnolum that were that were assumed to be associated pretty heavily with Eastern Hemlock. So we have those. There was a paper by Stevens and colleagues, excuse me, that also spoke about truffles and their connection to Eastern Hemlock. And a lot of them we just don't know. So I'm going to give a guess at least 7 to 8 that we know of, which seems like a small number. But again, we don't know everything right now, and there may be other species as well. Does that answer your question?
Yes, you said after this question. Perfect. Okay, I do have another question that's it's pretty basic. I wonder how does the diversity of the fungal community in Kejimkujik compare to other places? Is it, it's a protected area? Is it like more diverse because there's less disturbance? Is it less diverse for some reason? Yeah, I wonder.
Yeah, I can't fully answer that. Mostly just because excuse me again, mostly just because other areas haven't been as well studied as Kejimkujik. Obviously, being a national park, it's preserved pretty well. It's not as disturbed as some other forests, and it's protected. So I've seen high diversity there. I've seen, from a purely observational perspective, I've seen high diversity in the southern part of Nova Scotia as well. And in Cape Breton, when I've gone on hikes, your best bets for diversity tend to be, again, observationally. So if someone knows better than me, definitely take them over this. The best bets for diversity, in my opinion, seem to be those wild areas that have been not disturbed by human activity. But if we want to discover this more, I would definitely recommend going on iNaturalist and taking a look what other people have found, and also participating in MycoBlitz, because that'll get us lots more specimens, and you'll be helping poor master's students like me that just want data.
That's a great idea. We actually had Keith here on Monday that did a small MycoBlitz session with us with all staff. So that was a really great experience. And yeah, Keith was a wealth of knowledge, and I think we all learned an incredible amount of about fungi that day. Yeah, it's really cool.
Wonderful.
Chad, on your side of things, do we have anything else there?
No more questions.
No. Okay.
A few comments saying great presentation, great job, very interesting research.
Thank you. Thank you so much.
Yeah. Oh. Okay, Keith has one more thing coming up here. So there has been, Keith is saying there has been a lot of collecting over the years in the Kentville Ravine. Do you think those herbarium records could be useful?
Absolutely. Once the time has passed, you can't go back and get data from it. So having historical collections preserved in herbariums is so valuable, so valuable. Even in my own project, they're looking at the data that we have. I wish I had so much more. There are whole some years that weren't collected, or they didn't get this parameter. But having the data that we have is already such a gift. So yeah, definitely any sort of herbarium specimen can be used.
Keith is also saying great talk, and I can just echo that. I really enjoyed it, and I learned so much about fungi today, and the HWA, it's destroying this entire ecosystem. It's incredible. It's not only insects and trees that are possibly affected, but there might be fungi that are affected too. So yeah, again, great talk. And if we don't have any further questions, which I don't think we do, we can probably start wrapping up here. So thank you so much, Rebecca, and thank you everyone else also for coming and joining the talk today. At this point here, I would also like to thank our sponsor of the talk, the Region of Queens, for making this seminar possible. And as always, you can stay up to date with our seminars by following us on Facebook, Instagram, on all of our social media. And you can also rewatch this seminar here. If you chimed in a little late and you missed the beginning, you can rewatch the seminar on our Facebook Live or our YouTube channel. There's also going to be 2 more seminars coming up for MTRI this summer during August. For the next one, we'll be joined by Dr. David Shuttler, also from Acadia University. And Dr. Shuttler will be sharing his research on white-tailed deer and the pathogens that have introduced to the province. For instance, the brainworm that also is affecting moose populations. And he'll also be sharing some information on ticks and tick monitoring in the province too. And that seminar will be happening at the same time on Thursday at 7 p.m. till 8 p.m. And if you'd like to join, you can join us again on either Zoom or Facebook. And yeah, with this, I would like to end today's session. Thanks again, Rebecca, for the great talk. And yeah, good luck for the rest of your masters. I'm excited to see the final results. And I hope everyone stays well, and we get to see you all very soon again. So good night.
And with that, I would like to thank Luca for his amazing work. This will be his last seminar with us this summer, and it's a big loss. Luca has gone above and beyond with everything he's done. Thank you so much, Luca. And of course, thank you, Rebecca. Amazing talk. Thanks, everyone.
Thanks for the kind words, Chad. Everyone watching, you'll be in good hands with Chad from now on. Oh, that'll be great. All right. See you all soon. Have a good evening.
You too. Thank you so much.